Hello everyone. In this video we are going to talk about DNA structure. I am sure that we have already discussed the DNA structure in our previous units. Here is, as you can see, a core review, which is exactly the same as what we talked, and this is a part of our core regarding DNA structure. Do you still remember what was this structure? Pause the video and tell me what was the name of this structure. Okay, yes, you're right. This is one nucleotide. Do you remember what is this part? And this one. And this one. You can pause the video and think about. Yes, this was phosphate, this is sugar, and this is base. Do you remember the name of the sugar over here? Pause the video and think. Yes, it was ribose. But was it a ribose or something else? Do you remember? Yes, it was deoxyribose, not the ribose. Do you still remember what was the difference between ribose and deoxyribose? Pause the video. Okay. The structure of deoxyribose has a slightly one difference from the ribose, if you remember, and that was here. In that case, normally it must have OH as hydroxyl group, but it doesn't. So the hydroxyl group has become to deoxy, which means the oxygen has been removed and it has turned to H instead of OH. That's what the difference between the deoxyribose and ribose. Okay, all these are related to the previous course session, and then we have hydrogen bonds between the bases, and you see that there are in here we have 3 hydrogen bonds between 2 bases, and sometimes we have 2 hydrogen bonds. Pause the video and tell me what kind of bases can be here and what kind of bases here. Okay, I think you answered correctly. The bases that can be here, it could be either G, C, or the reverse, C here and G. Why? Because there is only between C and G that it is possible to have 3 hydrogen bonds. And here either could be A and T or T A. In both cases there are hydrogen bonds between these 2 bases, 2 hydrogen bonds between A and T, and 3 hydrogen bonds between C and G. Okay. In this slide, you will see the covalent bonds between phosphate group and the carbon number 3, which makes a very strong bond, and actually it is mentioned as sugar phosphate backbone, because phosphate, sugar, phosphate, sugar, phosphate, sugar, and it makes like a backbone of the DNA. That's why it is called sugar phosphate backbone. And the critical point over here is the number of the carbons starting from 1, 2, 3, 4, 5. So there is a direction, there is an order, and always the phosphate which is attached, the carbon number 5 actually is here. So the phosphate which is attached to carbon number 5 is always attached to carbon number 3. So this 5 to this 3, again this 5 to this 3, this 5 to this 3. So there is always and the last would be 3. So the direction of the DNA will be considered based on the last carbon which is remained open, and the last phosphate group which is also remained open, which is connected to the carbon 5. Because of that, in this case, the last one, which is free, is 3 prime. Yeah, and also they will change the name of the 3 by adding a prime, so that's why it is called 3 prime. And carbon number 5, and then because it is at the end of the DNA by ending to carbon 5, it is called 5 prime. So always there is a 3 prime at one end and 5 prime at the other end. Can you tell me what is the direction of 5 prime to 3 prime end? Yes, so this is the direction of 5 prime to 3 prime at this molecule. And there is always anti-parallel strands. DNA are anti-parallelic strands. Once the 5 prime to 3 prime is from one direction, this direction, the 5 prime to 3 prime for the complementary strand would be in the opposite direction. So 5 prime to 3 prime would be from here to here. So it depends on how do you want to interpret your DNA strand, if you want to talk about 5 prime to 3 prime or 3 prime to 5 prime. According to this slide, which is showing like this arrow is showing like 3 prime to 5 prime. Okay, so just forget about this arrow. Consider the arrow that I drew, because the direction which is important for us is 5 prime to 3 prime. So we always talk about the 5 prime to 3 prime. So that's why the arrow that we are always necessarily talk about is 5 prime to 3 prime direction, and the parallel one, the parallel strand, has the 5 prime to 3 prime into the opposite direction. Another explanation for the name of the bases are dividing them into purines and pyrimidines. Pause the video and tell me what is the difference between purine and pyrimidines. Okay, I'm sure that you already known what is the answer. Purines are those bases with having 2 rings, and pyrimidines are those bases having only 1 ring. How do you remember which one is purine and which one is pyrimidine? Okay, so there is a trick, and that trick is coming from here. This is pyrimidine, and this is thymine having Y, and this is cytosine again having Y. So thymine with Y, cytosine with Y, and pyrimidine with Y. So you remember that pyrimidines are thymine and cytosine that have Y, and the rest and the other 2 are adenine and guanine, which are considered as purines. This is the way that you remember, and you never forget which 2 bases are pyrimidine and which 2 bases are purine. And you always remember that G is matching with C, and the reason is because this is the way that you remember and memorize is because this G has like same way of writing as C, both have like a half circle. How is the double helix structure maintained? Well, this is a very nice slide, is explaining how a double helix structure having some special characteristics such as hydrogen bonds or other characteristics from the nitrogenous bases or the complementary base pairing or the sugar phosphate backbone, and all of these characteristics make a very unique structure, which we know as DNA structure. So let's see one by one and which each feature is making what kind of characteristics which is important in the DNA structure. Okay, first of all, hydrogen bonds. hydrogen bonds are not only between the bases, which are responsible for holding complementary base pairs, like here. So between C and G, or between A and T. Okay. So it is not just based on, it is not just responsible for making or for having the complementary strands attached to each other, but also there are hydrogen bonds that they hold the adjacent sections together. This area, as you can see, these are hydrogen bonds that creates this form of the DNA structure, which makes this curve, and the way that they are reaching to each other, and the way that these curves are creating this form inside the DNA molecule is because of the presence of hydrogen bonds between the adjacent sections. Let's say the next structure which is important is sugar phosphate backbone. Sugar phosphate backbone, as you can see here, it is hydrophilic. Why it is hydrophilic, do you remember? Yeah, because the structure of sugar phosphate had a phosphate group over here, and this phosphate group which was linked to a sugar makes it hydrophilic. Do you remember hydrophile? So phosphates are having hydrophilic structure, so that's why it is important for giving these characteristics to the backbone. Then what happens when there is a hydrophilic tendency? What happens? If you go back to the molecular chemistry of the molecules that we studied before, hydrophile means what? Do you remember? Pause the video and tell me what is hydrophile. Great. Hydrophile means they like water. Hydro means water and phil means they like it. So they like water. So hydrophilic structure means that they are liking water, so they are facing water molecules rather than ignoring. Another one, nitrogenous bases are very reactive, so are protected inside. So this feature that sugar phosphate backbone, because of the hydrophilic, stay outside. On the other hand, the nucleotides, the nitrogenous bases from the nucleotides stay inside because they are highly reactive and kind of protective by staying inside the molecule. So what are the other features? Well, you know that these polynucleotides are in the opposite directions. So that's another thing. They are antiparallelic. And this feature and this structure of the molecules, if you go back and see how the sugar backbone are having the numbers 5' to 3' in one side and 3' to 5' in the other side. I don't know why this pen doesn't work for me very well. I don't like it. Okay, it seems that it doesn't work in a small spaces. I should use it when I am making a rather longer lines. Anyway, so suppose that this is a 5' to 3'. I don't use this pen anymore. I don't like. Anyway, another thing, complementary base pairing which ensures that mistakes are not made when copying or transcribing DNA. And this is another feature. Once they have the 3 hydrogen bonds between C and G and A and T, they are unique, they are specific, and it doesn't allow mistakes. So all these features, including hydrogen bonds for keeping the adjacent molecules to keep this form of the curve, and also being a hydrophilic structure of the sugar backbone, and then the nitrogen spaces that they are having hydrogen bonds between themselves, which makes it protected inside, and also the antiparallelic direction, which is because of the 5' to 3' way of nucleotide while they're attaching to each other. All of these makes that unique structure of the DNA. Here is a good question. Outline the evidence that supports complementary base pairing. Nice. I'm using this time a box. Is it good? I don't know, maybe. Anyway, I catch your attention here. Outline the evidence that support complementary base pairing. Great. How do you want to explain that? How do you want to outline? You remember outline? Let me try this one. No, not this one. Oh, here, box. Okay. Do you remember outline? It's a very important word in IB examples. IB exams. So when we say outline, means you have to explain completely one by one and bring your results and deduction on the evidence of supporting complementary base pairing. So what do we have for outlining complementary base pairing? First, we know that the double helix DNA is tightly packed and regular. It is tightly packed and regular. This is very important. Ooh, sorry, I didn't know that that's gonna be something like this. It gets really big. Okay, so once again, because the DNA helix is tightly packed and regular, if you go back and see the 51 picture, famous picture from Rosalind Franklin from the X-ray diffraction, you see that there was a regular pattern that was like something like X, and it was completely a regular pattern that makes it something that you can say because of that regularity, then we can say that pyrimidines need to pair with purines. That's one outline. That's one outline. Because of tightly packed and regular, pyrimidines are paired with purines. Another one is because electrical charges of adenine and thymine are compatible, and the opposite. So they allow to make hydrogen bonds between them. So this is another outlining. Because these two nitrogen spaces are can structurally attach to each other and they are compatible, so we can, this is another reason that we can say that, oh yeah, this must be another evidence for complementary base pairing. And the third one also is explaining between cytosine and guanine the same way. These are also having a compatibility, or they're compatible because of having 3 hydrogen bonds that they can form to each other.